to the Western world. Leap ahead about 50, 60 years. You're now in the middle of the 20th century, the 1950, 1960. Now we have many more that are known at that time. In the plant area, you have DMT, for example. Found in many plants, DMT is not active orally, but it's certainly active in snuffings and injections using medical tools, inject smoking. Many plants are now known to have DMT as a principal alkaloid. Actually, DMT, dimethyltryptamine, was first synthesized in the early 1930s by Mansky in Canada. Had no idea of its pharmacology. Pharmacology was unknown. It's not until the early 1950s that it was found to be a component of one of the snuffs of South America. And from that, it suddenly became a point of medical exploration. There was the Stephen Zara, who is a chemist I know rather well, he's still alive, and NIDA, who went into this and synthesized said dimethyltryptamine, the diethyltryptamine, dipropyltryptamine, dibutyltryptamine, kept making the chains longer and longer. He discovered that the longer chain ones were orally active, and he explored 4 or 5 of these compounds and published on this information. He also made allyltryptamine, but he never explicitly told how he made it. There's an interesting story there, I'm going to get into it. But he had made the compounds, had found their biological activity. He found that anything larger than DMT was orally active, and as you got larger and heavier, you got less and less potent. So that was the first contribution synthetically in this whole area. There is the area that was talked about some yesterday of psilocybin, psilocin, the mushrooms, the active mushrooms, the shrooms as they call them in the United States in the slang, where you have the psilocybe cubensis, the work of Gordon Wasson of trying to bringing the mushrooms back into the United States from the Oaxaca area, the Ibero Indians, is right, of the Oaxaca area. And this led to work that was done, much of it was done by Hoffman, of the synthesis of psilocybin, of psilocin, and in Sandoz laboratories where we worked, they had made the diethyl, I believe the dipropyl, I know the diethyl homolog, and these materials are orally active, they're the components of the mushroom, and they had made the psilocin, which is the de-esterified compound, and the N,N-diethyl. Another plant source in this middle of this last century were what are called MGS, morning glory seeds. Morning glory seeds became quite popular. No one knew quite what they were. These from Mexico, the Ipomoea violacea. You have the baby Hawaiian red rose, Argyria nervosa. These are materials that contain alkaloids in them, but no one had worked out exactly what was in them and why they were biologically active. But a great popularity sprung forth in I believe it was the late 1950s, early 60s, of consuming handfuls of Argyria, spoonfuls of the Ipomoea, handfuls of seeds, maybe 100 seeds or 200 seeds for a trip. These are, plant sources. At this time, there were also a number of synthetic materials that came forth on the market, into the scientific community, I should say, in the area of psychedelic drugs. The major initiator of the synthetics were the Hoffmans worked on LSD. LSD was first synthesized, I believe, in 1938, and it was not known to be active. No searching for activity, looking for other things in Sandoz. And it was about 5 years later, in 1943, I believe it was, that Hoffman, they say accidentally, we don't know, he will not use any term other than accidental, got exposed to a small amount of it in him, and the experience, of course, is famous for the bicycle ride and the popularization and the making widely known in the scientific community and the medical community the intense potency of LSD as a compound. LSD is active, as you know, as many people know, 50, 100, 150 microgram levels. He had synthesized at that time about 4 or 5 other materials, the isomers that were not active, but he made the N-methyl, the 1-methyl, the 2-methyl, the 1,2 bromo compounds, that had biological activity, and perhaps there are a total of 4 or 5 compounds made at Sandoz that were known to be biologically active or had been explored biologically. Another material that was first explored in Canada, an outgrowth synthetically of mescaline, explored in Canada as ways of interfering with the visual effects of a spinning wheel that had lights and colors on it. And they tried to use this as a way of distorting the color sense, material called TMA, trimethoxyamphetamine. This is mescaline with a methyl group hooked onto the alpha position. So it's really an amphetamine chain, and it was called TMA by the people in Canada. And I had made the compound too. I called it TMA. In my case, I was looking at it as having mescaline-like action. And it is indeed about twice as potent as mescaline. And it's a basis from which I made a number of compounds. If one carbon, it makes it better, then 2 and 3 and 4 should be better and better and better. I made it out to about 8 carbons, and only the 1 carbon was the active one. All the rest are totally uninteresting. But anyway, TMA in this area was the first synthetics that followed the general area of mescaline. Also, a person down in Los Angeles at the University of California, UCLA, Gordon Alice, had made the compound, the basic amphetamine compound, with a methylene oxy group on it called MDA. And it was patented by, I forget the company, patented by someone as an appetite suppressant, which indeed it was, but it never went commercial because it turned you on. And that was not the thing you wanted a pharmaceutical company wanted their product to do. So the whole area was dropped. But it was patented as an anorexic, and it was a psychedelic known at that time. One other thing in the same area, about 1950-something, both Gordon Alice in Los Angeles and I had made a material we called MMDA, methoxymethylene-doxyamphetamine. The interesting story there is that he had made the material, I knew that, and he knew that I had made the material. We both had not determined its activity yet, but we were going to do so, and we planned to meet in January of the year. It turns out in the end of November the previous year, he died an unexpected death. And it died of a diabetes problem, and he was a pharmacologist who had worked out the use of insulin against diabetes problems in general. And that he should die of this problem, I found to be very, very challenging, very mysterious. Because he was also working on tryptamines, as I was working on tryptamines, and I had this fantasy that he had gotten into a tryptamine that's very lethal. And I would like very much to have met him before he died and find out what he was working on, so I could stay away from that particular area. And communication was never established. But we did have, he was, and I were the earlier studies of MDA, the methoxyamphetamine, and that's basically what was known in the 1950s. And total of all these compounds I'm talking about adds up to perhaps 20 compounds. So now let's skip ahead to the present day where we are now. We're now 50 years later yet. The number of plants have really become quite a bit more extensive. Not at the same growth as before, but still quite real. Many cacti have become active on the scene. Everyone knows, most people know about the many examples of upstairs, the well-known San Pedro, the Trichoceros pachanoi. There are many Trichoceros cactus that have been explored, most of them analytically and chemically, but some of them in human trials. And the number of cactus that contain mescaline is in excess of 50. There are actually 50, currently known about 55 cactus species that contain mescaline. Other cactus I've worked with in the outside of the Trichoceros. You have Trichoceros I saw upstairs, the Macrodonus. You have Terchechii, various variants of Trichoceros. Very rich. One of the Terchechii samples is a good one I got from northern part of Argentina, northwestern Argentina, in which the Terchechii has, I ran its analysis on it, the major alkaloid is N-methylmescaline, interestingly enough. A compound extremely similar, it has the same relationship